Welcome to Explainable TV, your one source to easily understand science, new inventions, and concepts in the most simple way. In today's episode, we're going to talk about COVID-19 vaccines, how they work, and how these were developed so fast. Before talking about vaccines, let's briefly discuss how our immune system functions. Once we have been exposed to an infection, our immune system kicks into action to fight the disease by producing antibodies. And even after recovery, our immune system, or the antibodies, remember the threat for some time. These antibodies are basically proteins that circulate in our blood, which quickly recognize and disable the invader upon contact, thereby preventing or minimizing chances of same illness for some time. This is why we mostly do not get sick with the same infection twice. Coming over to the second question of the day, that is, how vaccines work. Vaccines encourage our immune system to make antibodies against a particular disease without us having to go through that illness. In the 20th century, most vaccines took well over a decade to research, test, and produce. Traditional vaccines contain weakened versions of viruses or amputated sections of a single virus, both of which required time-intensive research to prepare. But the vaccines for COVID-19 cleared the threshold for emergency use in less than 11 months. So, how does COVID-19 vaccines work? And how these were developed so fast? The secret behind this speed is a medical technology that has been developing for decades, the mRNA vaccine. This approach offers significant advantages over traditional vaccines. It uses our body's existing cellular machinery to trigger an immune response, thus protecting us from viruses without us experiencing the actual infection. Moreover, mRNA vaccines don't actually contain any viral particles, so they don't have to be built from scratch. So how do these revolutionary vaccines actually work? The key ingredient is in the name. mRNA, or messenger ribonucleic acid, which is a naturally occurring molecule that encodes the instructions for producing proteins related to a specific antibody. When our cells process mRNA, a part of the cell called the ribosome translates and follows the encoded instructions to build the specified protein. Using this method, scientists safely introduce the virus to our body and the immune system. First, researchers encode trillions of mRNA molecules with the instructions for a specific viral protein. This part of the virus is harmless by itself, but helpful for training our body's immune response. Then, they inject those molecules into a nanoparticle, many times smaller than the average human body cell. This nanoparticle is made of lipids, the same type of fatty material that forms the membrane around our cells. But these lipids have been specially engineered to protect the mRNA on its journey through the body and assist its entry into the cell. Once injected, the nanoparticles disperse and encounter our cells. The lipid coating on each nanoparticle fuses with the lipid membrane of the cell and releases the mRNA to do its work. Once inside the cell, the ribosome translates the mRNA's instructions and begins assembling the viral protein. In COVID-19 vaccines, that protein is one of the spikes, typically found on the virus's surface. Without the rest of the virus, this lone spike is not infectious, but it does trigger our immune response. Activating the immune system can be taxing on the body, resulting in brief fatigue, fever, and muscle soreness in some people. But this doesn't mean the recipient is sick, it means the vaccine is working. The body is producing antibodies to fight that viral protein that will then stick around to defend against future COVID-19 infections. And since this particular protein is likely to be found in most COVID variants, these antibodies should reduce the threat of catching new strains. In fact, every mRNA vaccine has roughly the same list of ingredients, with some different configurable components. Imagine a reliable, robustly tested vaccine that can treat any disease by swapping out a single component. To treat a new illness, researchers would identify the right viral protein encode it into mRNA, and then swap that mRNA into the existing vaccine platform. This could make it possible to develop new vaccines in weeks, giving humanity a flexible new tool in the never-ending fight against disease. And in the future, this approach might be able to treat new diseases almost as quickly as they emerge. Thanks for watching. Please leave your feedback if you liked the video. Don't forget to subscribe and share with your friends.